Welcome to this particular module, and this is the last module for this particular lecture, which is on tissue and cell culture techniques. So, in the last lecture or series of 2 lectures actually, or 2 modules I should say, we have seen what is a cell, cell theory, incubator, biosafety hood, right, and how to use the biosafety hood from the video that I have shown you as a part of the module. Let us see today the few more equipment and chemicals that are required in this particular technique. Okay. So, if you see the slide, what I am showing you is a refrigerator, and refrigerator is not a general refrigerator that you can see at the home, but it is for maintaining the cells at 4 degrees centigrade, and as well as media, liquid media, growth factors, right, enzymes such as trypsin, but enzymes such as trypsin and media components like glutamine, serum, etc., we can use minus 20 degrees centigrade, right. So, the another point is not all the laboratories would have enough funds to maintain in this particular fashion. In that, a normal refrigerator you can use to store the freezer, you can use to store the enzyme and glutamine. Of course, you should make sure that there is no food along with the refrigerator, but as a alternative technique, if you do not have an exclusive refrigerator for maintaining the cells, you can use a normal refrigerator, a freezer, to keep your cells and the media alive and in a proper shape. But it is always advised to use the refrigerator that is meant for cells and tissue and for the media. Make sure that you do not have food along with your cells, do not have any kind of other stuff along with in the refrigerator. It is extremely dangerous to mix 2 things together. Bacteria, like I said, is extremely harmful. In any case, have a general practice if it is a wet laboratory where there are any chemicals or biological components or anything to do with any chemicals, you should not eat any food or drink water within the laboratory. Alright, make a practice. It is a good laboratory practices. It is a good it is wise in terms of how to work in a laboratory. So, if it is a dry laboratory where there are only systems, yeah, right, you may prefer to drink or eat depending how the lab works and how your PI allows you to work in the laboratory. But what I feel is in general you should avoid eating in a laboratory environment. Okay. In particular, for sure not in a wet laboratory. Alright. So, having said that, there are several tissue culture plastics, as you can see here, flask, 96 well plates, right over here, 96 well plates, 48 well plates, 24 12 6 well plates. There are petri dishes, and these will be treated with polystyrene. These are all treated with polystyrene. Okay. Let us see the another equipment, and that is extremely important equipment, which is our inverted microscope. Okay, it is called inverted biological microscope. All right. So, what the microscope will consist of? Microscope consists of a light source, condenser, objectives, you can change the objectives, and room for petri dishes like here, right, or tissue culture flasks. Okay. Then usually inverted microscope is used inside a tissue culture room to look at the status of the cells that are being cultured, right. You can see here the status of the cells, you can see here, and you can see here, right. If the cells are confluent, whether it has reached confluency, we can use inverted microscope to understand that. Different magnifications are possible from 10x to 100x. We use the microscope to see if the cells have become confluent, right, how the health status of the cells is, and when exactly the passage of the cells, when exactly to passage the cells for the next cycle. Inverted microscope gives a better access for imaging of live cells. Alright. So, we have a inverted microscope in a laboratory. What is the next equipment? Next equipment is our autoclave. We will see inverted microscope as a part of the lab component in detail. Okay. how to operate inverted microscope, how to load the slide, how can we see the cells, everything we will see as a part of the lab component. If you see autoclave, autoclave is used to sterilize the equipment, utensils, etc., that are used for tissue culturing for both reuse and safe disposal. So, you see whenever we work with any kind of equipment where we have used bacteria or cells, we and once the experiments are over, we have to sterilize this equipment, even you want to discard it in a biosafety bag. So, if you really see, it looks like a ultra modern pressure cooker, no. So, it is actually it is same, because it operates at a high temperature like 120 140 degrees centigrade, and you can see there is a pressure gauge, there is a regulating device, safety valve, autoclave lid, and there is a autoclave body, there is a vacuum release and steam release valve, there is a auto stand, and this actually boils down the bacteria or kills the bacteria by boiling it up, right. If you see, I do not know how much of you guys know the how to purify the water, or once upon a time in early 90s, not every place in our country where was facilitated with a clean drinking water. I am sure still it is not, okay. But the how to clean the water when the water is muddy, but it is a drinking water. So, what people used to do is they used to boil the water, and when you boil the water, then the germs within the water will die. Same thing, if I boil, if I boil the if I place the utensils in a boiling water, right, what will happen? The germs would die, right. That is called sterilization. You make it sterile, kill the bacteria, kill any contamination from the device or from the equipment or utensils that we used in the biology laboratory, right. So, like I said, it is like a high tech pressure cooker, and it removes microorganisms. Actually, it kills, it is not just removing, it kills like virus, bacteria, fungus, spores, etc., using high pressure and high temperature steam sterilization. This is another equipment very important, and again you will see as a part of the lab component is the micro-manipulator. Micro-manipulator. So, what exactly micro-manipulator means? Micro-manipulator is an equipment that is used to physically interact with a sample under study, and preferably under the microscope. Using specially designed needles, mechanical forces with high precision and accuracy can be applied to a sample, and its response can be studied using sensors placed below the sample holding stage. The system is used to study the stiffness and the material properties of biological tissue, as well as it helps define the exact step sizes of XYZ moments, which can also be programmed to definite time steps. Easy? No, it is confusing, right. So, so what does that mean, right? Micro-manipulator is used. So, let me give you an example, it becomes easier for you then, okay. I have a tissue here, right, this is a tissue. I want to press this tissue. Right, this is my indenter, it will indent, it will press, right. Now, let us assume the tissue is just 10 microns, 10 micrometer. Our hair size, human hair size varies from 80 to 100 micrometers, the thickness 80 to 100 micrometers. What is the tissue thickness? Only 10 micrometers, only 10 micrometers. So, if I want to understand the stiffness of the tissue, how much stiff my tissue is, if I want to know the stiffness of the tissue, what will I do? I will indent the tissue, I will press the tissue, and I have to indent the tissue in by going down few microns in z direction, right. So, in z direction, if I press the indenter and I have to go down few microns, I have to use a indenter. But how can I control few microns of pressing the tissue. Few microns is possible with the help of a manipulator, and that manipulator is my XYZ manipulator, is also called micro-manipulator, and that is why it is called micro-manipulator. Okay. It can control the movement of my indenter in Z, in X or in Y, right, in terms of with a precision of few nanometers actually, but step size of few microns. We can it is really accurate. So, you have to understand three different things. It is your homework: resolution, accuracy, precision. What's the difference? Is resolution and accuracy same? So, just go and understand these three terms and you will understand. So, my point is, if I take this as a tissue, right, and I have my this is the indenter. If I want to press the tissue, how can I press it with micron accuracy? I can only press this with micron accuracy when I have connected my indenter to a micro manipulator. That is how I am connecting micro. Now, another thing that is written here is that when you press the tissue, you can place the tissue using sensor placed below the sample holding stage. Where is the sensor here? What I will do is, if I, so let us understand one more term before we understand this thing. There is a term called piezoresistivity. What is called? Piezo, P-I-E-Z-O, resistive, R-E-S-I-S-T-I-V-E, piezoresistive. When we apply pressure, there is change in resistance, that is a piezoresistive. So, suppose I have a sensor on which this is my sensor, let us say, right. This is my sensor on which I have placed the tissue, right, and now I am indenting the tissue. I am pressing the tissue with the help of indenter connected to my macro manipulator. When I press the tissue, what will happen? Because of the stiffness of the tissue, the force applied at the tip or the top of the tissue will be transmitted to the bottom of the tissue depending on the stiffness, right. It will not be same force. So, whatever there is a change, whenever I apply a force, there will be change in the resistance of the sensor. Why? Because it is a piezoresistive sensor. So, when the piezoresistive sensor is there, when you apply a force, there is change in resistance, and that change in resistance depending on how much force is transmitted from the indenter to the sensor, and that depends on the stiffness of the tissue. You got it? So, for example, if my tissue is stiff and if I apply a force, the force reaching to the sensor would be different compared to when my tissue is more elastic, right. So, the elasticity or a mechanical property of a tissue can be identified with the help of a piezoresistive sensor on which there is a tissue placed, and by pressing the piezoresistive sensor with the help of a indenter, and this you can do with micron accuracy with the help of micro manipulator. That is the role of micro manipulator in a laboratory where we to understand the tissue. It is very important, because when you or when the cancer progresses, there is a change in the tissue property, right. It is not just the constituent of tissue changes, right, but what we have is all the gold standard which is right now used, which are all biomarkers, H&E biomarker, postrogen biomarker, estrogen biomarkers, lot of biomarkers are there, right, HER biomarkers. But can we come up with an idea that if there is a change in the, or if there is a progress in the cancer, right, there is a change in the stage of the cancer, the tissue property should change. And if we can use one example, which is stiffness, cancerous tissue would be more stiffer compared to the normal tissue depending on the stage of a tissue, or vice versa, right. So, if there is a change in the tissue property, that is what my point is. If there is a change in the stiffness of the tissue property as the cancer progresses, can we use the same platform micro manipulator with a indenter and a piezoresistor on which there is a tissue placed to understand the stiffness. So, if we can understand stiffness, from the stiffness value we can say that this particular tissue from the patient belongs to this particular stage of cancer. For example, if I give you a very simple example, if it is from normal region or benign, or it is from DCIS, which is ductal carcinoma in situ, or it is invasive carcinoma in situ, or sorry, it is invasive carcinoma. So, or invasive ductal carcinoma, I mean, which is IDC. So, it is like benign DCIS or IDC, then depending on the stage the tissue property should change, and that is what our idea is. How can we measure this change in the tissue stiffness with the help of a given sensor, right, and can we develop a system around it, that we will see as a part of this course. All right. So, this is a micro manipulator that we have seen. Let us go to the next slide. The next slide is Microtome, and this tool is used to cut extremely thin slices of material called sections. Right. They are used in microscopy and tissue culture, allowing for the preparation of samples which can both be used for observation under light or electron microscope. So, if I take out the tissue from the patient, let us say using biopsy BIOPSY, I am extracting the tissue from the patient using biopsy, and then I have to slice the tissue. How can I slice the tissue? I can slice the tissue with the help of microtome. You see, create thin slices of material, right, you see here, called sections. They are used in microscope and tissue culture, allowing for the preparation of sample which can be used for observation under light or electron microscope, and further for culturing. The method itself is called microtome, and that is why we have microtome. The section thickness can go as low as 50 nanometer, with average sections being of 100 microns in thickness. About 100 microns is the thickness, average thickness when we slice the tissue, and like it is written, it can be used to understand the change in the properties of tissue under the light or electron microscope, and it can also be used for further culturing the tissue. So, if you the equipment looks as shown in the slide, it is a drive wheel, it is a block holder, a paraffin block, a tissue, and a still knife. So, when you want to store the tissue for long time, you can paraffinize the tissue. Paraffin block is there, that is why it is written over here, and when the tissue comes to your laboratory, if it is paraffinized tissue and you want to deparaffinize it, so that means you have to take out the tissue from the wax, then you can use xylene, XYLENE, xylene. Okay. So, xylene is used to deparaffinize the tissue from the paraffin block. So, paraffin block is used to store the tissue for a long time. So, if I play this one, then you will understand the details about how the microtome can be used. Look at the video and let us see the next slide later. Now, let us go to the next set of equipment. This is centrifuge, and a centrifuge is used to concentrate the cell suspension and remove the supernatant. So, for example, if I have a tube in which I am placing the cells along with media, right, like this. Now, my idea is that I want cells to be only at the bottom of the tissue, and remaining thing should be on the top here, cells should be at the bottom. What can I do? I can centrifuge this particular tube at high RPM, right, just to separate or remove the supernatant. Then this supernatant I will just throw it out, and I will have only cells left at the bottom of my tube. This thing we can do with the help of a system called centrifuge, and the process is called centrifugation. All right. So, the equipment is used to concentrate the cell suspension and remove the supernatant. The equipment has controls to set the required RPM and time of rotation. Depending on how fast you rotate, how much time you take for rotation, we can separate the suspension from the cells. We can insert multiple Falcon tubes. These are called Falcon tubes, and you can see there are array of Falcon tubes right over here. At the same time, a sample video showing the operation is shown right over here. So, let us see how the centrifuge works, right, in real time, and then we continue to the next slide. Okay.
The Executive Series Centrifuge is the newest addition to the elite product line of Insight Global Biologics. And taking advantage of all aspects of superior separation, the Executive Series Centrifuge comes fully equipped. A high-performing machine with an enhanced look and smaller footprint is now here. When looking at the Executive Series Centrifuge, you will first notice its large multifunction buttons and clear display. To the left is the time button, where you will turn the dial to set the time and press to start and stop centrifugation. To the right, you will find the speed button, which also can be turned to set the RPM speed, and press to toggle between the RCF and RPM display. These buttons are clear and easy to operate. The centrifuge comes with the controlled deceleration and soft braking feature. This minimizes cell agitation and resuspension during processing. Optimized separation is achieved without activation, providing the best achievable cell concentration for your procedure. To activate this feature, press and hold the short button until you see the BR off indicated on the display. This machine has an automatic lock and release function for lid operation. To open the lid, power up the machine and press the open button. You can manually open the lid without AC power by releasing the pull string at the base of the machine, where you can pull to open. The lid automatically locks when closed. Looking inside, you will find that the machine comes with the proprietary rotor and two customizable swinging buckets. These buckets are designed to swing horizontally, then smoothly present to a full vertical position when completed. This prevents the buffy coat from resuspending into the plasma and prevents the platelet slip phenomenon, which degrades the quality of the final product. With these new concepts in platelet and bone marrow separation, the Executive Series Centrifuge now makes its mark as it enhances the quality of these products. Insight Global Biologics remain committed to be a market leader in regenerative biologics.
Okay. So, now if you see this particular slide, what do you see here? This slide shows the cryovials. Cryovials are the specialized vessel designed for storing biological material, human or animal cells at temperature as low as minus 196 degree centigrade. These are all cryovials, exclusively made for storing the cells at extremely low temperature, like cryopreservation, right. Now, there are some cautions and or you can say the rules of how to use the cryovials. The first one is that you have to use only in the gas phase of liquid nitrogen, and this vials should be used for cryopreservation, which is a process by which the biological material like cell tissues or even organs or organelles within the cells are susceptible to damage caused by unregulated chemical kinetics are preserved by cooling at very low temperature, right. We have seen cryopreservation, it is used to store the cells at low temperature, thus by preventing any kind of unregulated chemical kinetics. Also, we have to use solid carbon dioxide minus 196 degree centigrade liquid nitrogen. So, because this causes the unregulated biological activity to almost cease, right. Why you have to use minus 196 degree centigrade liquid nitrogen or minus 80 degree solid carbon dioxide? Because at this temperature the cell activity, which is unregulated biological activity, ceases down, and thus we can use the or we can preserve the cell for a longer time. The process of bringing the cells or reviving the cells or reviving the cells is called the thawing. Thawing, where we are bringing the cells back for our experiment from the cryopreservation back to the laboratory environment, and this the process is called thawing. Cool. So, cryovials are used for what? For preserving the tissue or preserving the cells, and what temperature it should be used? It should be used at minus 80 degree if you are using solid carbon dioxide, or minus 196 degree if you are using liquid nitrogen. The cryopreservation is used for, why it is used? Because it seizes the chemical kinetics and preserves the cell. How can we revive it? We can revive it by the process called thawing, right. So, this is what we have seen. Now, let us see another very important nutritional requirement for the cells to be alive, and that is cells have a complex nutritional requirement that must be met in the in vitro culture environment. You see how we can keep the cells alive and we can also grow the cells in a laboratory environment. So, for doing that, historically scientists used chick embryo extracts, plasma, sera, etc. as growth media. So, this is what is growth media. Okay. However, they varied in growth promoting characteristics and hence affected the reproducibility of the experiment. Every time it was not reproducible. So, today a number of chemicals defined formulations are available that suppose the consistent and reproducible growth of several primary and cell lines based cultures. Some of the chemicals that are used in the laboratory are Eagle's basal media, Eagle's Eagle's minimum essential media, which is called EMEM. We also use Delbaco's modified essential medium, which is called DMEM, which is extremely widely used, and then we have Iscovo's modified Delbaco's medium, IMDM, and finally we have Ham's F12, etc. Right. So, but in most of the laboratory you will see DMEM as a media which is used for preserving the cells or growing the cells, as discussed earlier. The various nutrients required for the cells are glucose, fats, fatty acid, lipids, phosphates and sulfur phosphates, ATP and amino acids, vitamins, minerals and sometimes antibiotics to prevent the growth of unwanted microorganisms. So, this is the combination of the media. Another major constituents of media is serum. It provides various growth factors, hormones, cell adhesion factors and other essential factors required for or required by the mammalian cells, right, for their long term growth and metabolism. Some common serum used are FBS, FCS, CH, HS and HOS. Now, what is the pH of the cell? The pH or optimum pH of the cell is from 7.2 to 7.4 and phenol red is used as an internal indicator. As cells consumes the nutrients, the pH of the medium will change. When the pH of the medium changes, the color of the solution also changes, giving indication that it is time to change the media, right. So, when the constituents of the media is consumed by the cells, then what happens, that the color of the media changes, and at that point we can see by looking at the color of the media, it is time to change the media, got it. So, this is the use of the media. Now, let us see what we have to show it to you or to offer you as a part of this particular this particular course, and this is the laboratory which we will show it to you, and we will show the details how the different things are used in the laboratory. As you can see, this is just a setup for developing a electronic nose, nose. We will discuss sometime if time permits, right, and the idea is that can we understand the disease from breath signature, and for that how can we develop different sensors. You can see here we have a stereo microscope, we have a metallurgical microscope, then we also have a inverted microscope. So, this is an inverted microscope, inverted INVERTED, inverted microscope. This is metallurgical, METTAL, metallurgical, sorry, METALLURGICAL, metallurgical microscope. This is stereo microscope, STEREO, stereo microscope, metallurgical microscope, right, and we have inverted microscope. We will see each in detail when we see the laboratory as a part of laboratory class. And then we have O1, you can see here we have the autoclave, you can see here we have biosafety hood, you can see here we have to store different antibiotics right over here, we have a CO2 incubator right over here. These are all positive pressure modules with HEPA filters. HEPA, HEPA. HEPA stands for high energy particulate air. High energy particulate air filters are used to create a controlled environment, which is class 100, class 1000, class 10,000 and so on. So, there are different class, clean room class, we will see the different clean room class in one of the lectures, right. And if you want to know more, you can just go to my lab website and you can see what are the facilities that I have in my laboratory, or instead I should say we have as an institute, as a department and as an institute for our students and for people who are interested to collaborate with the laboratory. Okay. So, having said that, if I go for another few of the things that we will discuss, these are only few. Okay. We have many, I have just put few just to make you understand. We will discuss about autoclave, that we have already seen. Then we will go for sonicator, how to use sonicator. We will see inverted microscope, hot plate stirrer, LCR meter, we will see biosafety hood. This is biosafety hood 2, level 2, CO2 incubator, O1, microcentrifuge, and we have vacuum pump. These are, like I said, is very few of the equipment right over here. I will show it to you many more as a part of the lab. So, so this is the existing infrastructure, and in the and you can understand that or let us conclude here because I think that the next class should be more focused on clean room technologies and what are the clean room and how the environment in the clean room affects the fabrication of a device, and why we require a clean room, right, and we will discuss more on that. But as far as the cell and tissue culture is concerned and the application is concerned and the equipment used for maintaining the cell and tissue cultures are concerned, this is the end of the this particular module and this particular lecture as well. So, this lecture I have divided into 3 different modules. If you see all 3 modules, you will have a basic idea of how the tissue and cell culture laboratory looks like, what are the equipment within the laboratory, and in next class let us see what we learn as a part of this particular course. which will be more on clean room fabrication and then design of electronic systems for cancer diagnosis, right. Till then you take care, I will see you in the next class.